Good afternoon. It's just wonderful to see all of you, on this bright, sunny day. And it is wonderful to have you come and listen to a very distinguished speaker. For those who don't know me, my name is Erasmus Okine. I'm the Vice President Research at this university. And I'm very, very happy to introduce our lecturer for the gairdner, hosted by the University of Lethbridge. This is the third gairdner lecture hosted on our campus, and I'm very happy to see so many faculty, staff, and students, colleagues, and some community members taking advantage of this opportunity. Indeed, I am excited, and I hope you are excited about today's presentation. The U of L is honored to have been selected again to host this lecture, and I'm very proud to welcome Dr. Lynn Marquardt, a 2015 Canada Gairdner International Award recipient, to our campus to share her experience. For those of you who don't know, the Gairdner Foundation recognizes excellence in health research. Their president, Dr. John Dirks, could not be here today, but did ask me to bring greetings and share a little background on the organization. The Gairdner Foundation, now in its 56th year, recognizes excellence in scientific endeavor that leads to the greater good of society. The awards are distributed, and they are only the Canada's globally known science prizes and include the Canada Gairdner International Awards, the Whiteman Award, and the Canada Gairdner Global Health Awards. These are our Canada's most prestigious awards in biomedical research and rank among the most honored scientific awards in the world. Indeed, nearly 30% of the Gairdner Award winners have gone on to win Nobel Prizes for their research. So who knows, we may have a future Nobel Prize winner amongst our midst today. The work of Gairdner Award winners forms the backbone of many medical and pharmaceutical advances that save lives and improve global health. Their breakthrough discoveries and innovations have led to CT scans, MRI imaging, PET scanning, the eradication of smallpox, drugs to permit organ transplants, and the HPV vaccine against cervical cancer. As you can see, the Gairdner Awards recognizes exceptional work taking place around the world. Moreover, they bring past and present award winners to campuses across the country as part of the national program we are part of today. Indeed, the U of L is one of the only 22 avenues in Canada hosting a Gairdner event this year, and only one of the 16 universities who had the opportunity to offer a high school lecture like we did this morning. Lynn, I understand that there were about 100 high school students who listened to you today, and I heard they were very enthused and excited to hear you speak. Indeed, it is an honor for the U of L to host this event here on our campus and to be able to extend the excitement around biomedical science into our community. I would like to take this opportunity to thank some of the sponsors who made this possible. The Government of Canada, the Government of Alberta, the Canadian Institute for Health Research, the Canadian Foundation for Innovation, the Burroughs Wellcome Fund, the Globe and Mail, and London Drugs, who sponsored the high school component of today's event. It is now my pleasure to introduce today's speaker, Dr. Lynn Marquardt. She is the director of the Center for RNA Biology, from Genome to Therapeutics, and professor of biochemistry and biophysics at the University of Rochester School of Medicine and Dentistry. As many of you know, RNA research is one of the fastest growing fields in the life sciences, and Dr. Marquardt is at the very forefront. In 1981, not many years ago, Lynn, she published a seminal paper in the journal Cell on the inherited disease beta-0 thalassemia, showing that the disease results from rapid degradation of beta-globin mRNA. Since then, she has continued to investigate the underlying mechanisms for mRNA degradation. She's won so many awards, but enough said about her already. Please let us welcome Dr. Lynn Marquardt to the stage.
Oh, yeah, take it. In fact, I think we can remove this too, right? Yeah? Great. Well, thank you, Represses. It's a pleasure to be here, and it's a tremendous honor for me to be receiving International Award. All right, so I'm going to talk about science now, and I encourage you to ask questions while I speak so that we can all stay on the same page. I'm going to talk about nonsense-mediated mRNA decay and its connection with human diseases, and how NMD, which is how we abbreviate nonsense-mediated mRNA decay, is a genome guardian and executor. So as I said, NMD stands for nonsense-mediated mRNA decay. And by way of review, I think you all know that translation normally terminates at one of three nonsense codons, which are shown here in red. These codons usually don't encode any amino acid, and instead they signal the release of the nascent protein and ultimately dissociation of the two ribosomal subunits from the mRNA template. Many years ago, we realized that a number of human diseases could be due to frame shift or nonsense mutations that result in the premature termination of translation. The disease exists because there's an absence of full-length functional protein. We also realized that if translation termination is sufficiently premature, then the mRNA will be targeted for NMD. And as you just heard, the first demonstration of this was a Cell paper in 1981, unstable beta-globin mRNA in mRNA-deficient beta-0 thalassemia. It's a hemolytic anemia. And then we went on to discover the same thing in another anemia, which is also a neurologic disorder, triose phosphatidylserine deficiency. This is a deficiency in a glycolytic enzyme. And that was work driven primarily by a graduate student in my lab at the time. NMD is a really important pathway because the truncated proteins that would derive from an mRNA that prematurely terminates translation can be toxic. And so it behooves the cell to degrade mRNAs that have the potential to encode a toxic protein. I think you all know diseases are heterogeneous at the molecular level. And NMD targets a third of all inherited diseases and a third of all acquired diseases like cancer. And some of the inherited diseases I've listed are cystic fibrosis, Duchenne and Becker muscular dystrophies, in addition to the beta-0 thalassemias that my lab has worked on and triose phosphatidylserine deficiencies. And then this is a very incomplete list of genes harboring nonsense mutations that generate premature termination codons, PTCs. So this is an incomplete list, and it is limited to nonsense mutations and not the frameshift mutations that can also generate a premature termination codon. And you recognize many of them, and I actually limited the list to those where I could fit in the name of the associated gene into this little column here. Notice tumor suppressors are also disease-causing because they can contain a premature termination codon that triggers NMD. This is a very simple overview of some of the nuclear and cytoplasmic steps that are involved in the expression of mammalian genes that encode proteins. Just by way of review, I think you all know that genes are transcribed in a collinear fashion into a precursor to mRNA, a precursor to messenger RNA. And then this pre-mRNA is processed both co- and post-transcriptionally. So the resulting mRNA lacks the introns. They're also called intervening sequences that are shown as lines in the pre-mRNA. And the mRNA also contains a cap at the 5' end and a 3' poly A tail at the other end. This mRNA is then exported to the cytoplasm where it directs the synthesis of proteins in a process called mRNA translation. And then each mRNA has an inherent half-life that can be regulated. So our studies of NMD have uncovered remarkable links between pre-mRNA splicing in the nucleus and mRNA translation and degradation in the cytoplasm. And we now know that these links are provided by what we call exon junction complexes, or EJCs, that are deposited a particular distance upstream of the exon-exon junctions, shown here as the vertical lines, that are generated by splicing. And these EJCs stably contain some of the NMD factors that we named after their orthologs in Saccharomyces cerevisiae as UPF factors. This was work done largely by Hervé Leyer when he was a postdoc in the lab, and we were working in collaboration with Melissa Moore, who was at Brandeis at the time. And then we also uncovered a new template for protein synthesis that we call the Pioneer Translation Initiation Complex. It turns out that NMD targets newly synthesized RNAs that have yet to lose the cap-binding protein heterodimer that's called CBP80 and CBP20 that's acquired by pre-mRNA, co-transcriptionally. And then if the mRNA derived by splicing, that Pioneer Translation Initiation Complex will also contain the post-splicing EJCs, or exon junction complexes. Even though we discovered NMD by studying human diseases, it turns out that NMD functions to downregulate abnormal transcripts that we generate all of the time. And this is because we have only 20,000 genes that encode proteins, but we need a lot more diversity than there being one protein generated per one gene. And part of the way we generate protein diversity is by alternative splicing. So you can generate more than one protein per gene. And this alternative splicing is regulated in a developmental and cell type specific way. Well, it turns out with that flexibility come a lot of mistakes. And a third of all spliced mRNAs produced in our cells are mistakes that are eliminated by NMD. I want to give you an example of how NMD downregulates alternatively spliced human transcripts. And in this example, the alternative exon is the red exon. And in this scenario, the normal splicing reaction, the productive splicing reaction, would splice out the red exon so that translation would initiate and terminate normally, no NMD. But if that exon were left in the mRNA and the exon had an in-frame termination codon, that would most likely result in NMD. Alternatively, if that exon were left in and it was not a multiple of 3 nucleotides, that would result in a shift in the reading frame and the generation of a premature termination codon downstream of the shift. This can be viewed as a type of intron retention. And NMD can be generated or targets for NMD can be generated because of alternative splicing events that lead to exon skipping as well. So you can imagine if an exon were skipped that was not a multiple of 3, it was in the coding region, you would generate a shift in the reading frame and a premature termination codon. And you know, there's nothing special about a premature termination codon that triggers NMD and splicing downstream of the normal termination codon, which could be converted to the type of codon that triggers NMD. So that's another way by which alternative splicing can lead to NMD. We've done work with a Canadian colleague, Ben Blenko, who's in Toronto, and shown that these NMD targets that are generated by splicing are largely mistakes. And we know that because they are not produced in a level that's constructive, high enough to change cell physiology, and also they're not conserved in a cell type or tissue-specific type way, which are hallmarks of constructive splicing, alternative splicing. And as I've said, the purpose of NMD is to downregulate mRNAs that have the potential to produce a truncated protein that's toxic to cells. And I want to give an example of that, but first I need to tell you that therefore NMD is viewed as a type of mRNA quality control pathway. It's also viewed as a type of mRNA surveillance. So the ribosome in the process of translation surveys the RNA to determine whether it should be degraded or not, depending upon where translation terminates, largely relative to where splicing occurred in the pre-mRNA that generated the mRNA. All right, here's the example of cell toxicity that occurs when NMD fails to occur. And the example derives from nonsense codons, premature termination codons, in the last exon of the human beta globin gene. These result in a dominantly inherited form of what is usually a recessive disease, thalassemia intermedia. Here's a picture of the normal human beta globin gene. Translation initiates in the first exon and terminates in the last exon. And this is the location of a premature termination codon that will not trigger NMD. You'll see why it doesn't in a minute when I show you rules. Turns out the beta globin mRNA that contains that premature termination codon has a near normal half-life. It's stable. But instead of producing full-length beta globin protein that can form a tetramer with alpha globin and generate functional hemoglobin that carries oxygen in the blood, the truncated protein that is produced can only dimerize with alpha globin. And that results in ineffective erythropoiesis and a dominantly inherited form of the disease. Truncated proteins can function in dominant negative ways. We were really interested in understanding how cells differentiate termination codons that trigger NMD from those that don't. What we did was we moved introns around in genes, and we also generated premature termination codons at different positions in genes. And we established the 50 to 55 nucleotide rule. That rule says if there is a premature termination codon in the purple region, which is more than 50 to 55 nucleotides upstream of that last exon-exon junction, then there will be NMD. But if the premature termination codon resides elsewhere, then there will be no NMD. And so you can see why PTCs in the last exon of the beta globin gene don't trigger NMD, just based on this rule. And remember how I told you there's an exon junction complex upstream of exon-exon junctions? And I'll show this more clearly later, but we established this rule before we knew about exon junction complexes. We proposed that they were marks. There must be some mark that's deposited on newly synthesized mRNAs that persists until the first round of translation. And this rule makes sense now that we know where the mark, the EJC, is, because if a ribosome translating the mRNA got any closer to the junction, it would physically remove the exon junction complex, and the mRNA would be immune to NMD. There isn't anything special about this last exon-exon junction. We think that generally exon-exon junctions, in fact, Hervé in his own lab showed approximately 80% of exon-exon junctions, or 80% of exon-exon junctions have an EJC. It's just that if you want to determine the boundary in your favorite mRNA, between PTCs that trigger NMD and those that don't. If you look right here, as you would predict, mRNAs that don't derive from splicing but have a premature termination codon are largely immune to NMD. And we've also shown that if you have a PTC here and reinitiate before the next exon-exon junction, depending upon the efficiency of reinitiation, NMD will be inhibited. Okay, now that we know the rule, let's go back and talk about how NMD can protect cells from potentially deleterious truncated proteins. Even when disease results from NMD-induced protein deficiency, the disease phenotype may be milder than and different from that caused by an expressed truncated protein. So NMD protects many heterozygous carriers of PTC-containing genes from manifesting a disease phenotype that would result if that truncated protein were expressed. This is a really short list. It's a very long list. This is just a piece of the list showing how, I've already talked about thalassemia, for example. If you have a PTC 5' to that boundary, you have a recessively inherited form of the disease. But if the PTC were 3' to the boundary and therefore not triggering NMD, dominantly inherited form of the disease. And you can go through the list and see how more severe the phenotype is if the PTC were positioned within the mRNA so that it failed to trigger NMD. All right, so we have NMD protecting our cells from mistakes that are made all the time. NMD has also been usurped by cells to function in important feedback mechanisms that include the coupling of NMD with alternative splicing. This is a mechanism that's used by many RNA binding proteins, and I'll give you one example, the polypyrimidine tract binding protein. This is the pre-mRNA from which PTB derives, and normally exon 11 makes it to the mRNA, so full-length PTB is generated. But it turns out if the cell makes too much PTB for the circumstances that it is subjected to, then PTB will feed back on the splicing of the pre-mRNA from which it derives. Remember, it's a splicing factor, and it will cause exon 11 skipping so that exon 11 doesn't make it to the final message. And exon 11 is in the coding region, not a multiple of 3 nucleotides. There's a shift in the reading frame and the generation of a premature termination codon in exon 12. That triggers NMD. So this is a way for this particular RNA binding protein and many others to auto-regulate by regulating the splicing of the pre-mRNA that encodes them. And then a third purpose of NMD is manifested by the fact that about 10% of our normal mRNAs, our physiologic mRNAs, are NMD targets. So this means NMD functions beyond being a quality control mechanism and beyond working in conjunction with alternative splicing. NMD serves as a cellular adaptive mechanism where cells regulate the efficiency of NMD. They usually downregulate the efficiency of NMD so as to change constructively to their environment, which itself is constantly changing. What do these normal NMD targets look like? Well, they can be mRNAs that have an upstream open reading frame. So they're going to terminate before, in the case of myogenin mRNA, the part of the mRNA that encodes myogenin is preceded by an upstream open reading frame that ends with a termination codon. And you can imagine if the reinitiation efficiency is low and there's a splicing event downstream of the open reading frame in the myosin encoding part, that mRNA will be a natural NMD target. There are mRNAs that naturally contain one or more introns within the 3' and translator region, so the normal termination codon triggers NMD. And then we've done a lot of work on selenoprotein mRNAs. These are specialized mRNAs that have a seleноcysteine insertion element in the 3' and translator region. Seleноcysteine is the 21st amino acid, and it is encoded in these specialized mRNAs by UGA, which is one of the 3 termination codons, nonsense codons that I talked about in one of the first slides. And we've fed rats increasing levels of selenium in their diet. And while increasing selenium levels will lead to more full-length spindle protein synthesis, there's always some level of NMD for these mRNAs. I want to show you some data, and this is work that we recently published, providing evidence that the efficiency of NMD can be regulated, in this case down-regulated by cells, as a means to adapt to changes in the cellular environment. And the stimulus, the change that I'm going to talk about, is the change brought about by DNA-damaging reagents that activate the p53 tumor suppressor pathway. And when this pathway, the DNA damage, is severe enough, cells will apoptose. So it's programmed cell death. This is work done by Max Popp, who is a postdoc in the lab. And we published this this year. Attenuation of NMD facilitates the response to chemotherapeutics. So frontline chemotherapeutics like doxorubicin damage DNA. And this is what one of the damaging reagents that Max studied. But here's the model. The model is that NMD is the molecular valve, and that can be tuned by the cell. So normally NMD is fully functional for that cell type, and what makes it to the cytoplasm and on polysomes to make proteins are mRNAs that are not NMD targets, either directly or indirectly, but they are not regulated by NMD. However, in the case of the DNA-damaging reagent doxorubicin, the chemotherapeutic, there is a downregulation of NMD because the key NMD factor, UPF1, and I'll explain that, is caspase cleaved. Do we have a lot of background noise? No? All right. And of course, that's going to inhibit NMD. So what makes it to the cytoplasm and on polysomes to make proteins are direct and indirect NMD targets, as well as unregulated mRNAs. And those direct and indirect targets promote cell death. They are pro-apoptotic. All right, let me show you some data. These are HeLa. They're human cells where we can regulate any gene that has a TET-responsive promoter. And we've driven the human beta globin gene that is PTC-free or contains a PTC that triggers NMD using the TET-off promoter. And when you block the production of mRNA by shutting off its promoter, one can watch how the RNA degrades. And you can see the PTC-free, the normal mRNA, doesn't degrade as a function of time. But there's NMD, and this is a biphasic decay curve because NMD is largely restricted to newly synthesized RNA. And then when NMD is over, the half-life of the mRNA is normal. It's the stuff that escaped NMD that has a normal half-life. Well, doxorubicin inhibits NMD. And puromycin, a control, a translational inhibitor, because NMD depends upon translation, puromycin also inhibits NMD in these cells. Well, we knew for many years that when we treated cells with cyclohexamide, for example, the key NMD factor UPF1, which is shown here in a Western blot, is not only full length, but we see these shorter products. And Max, being a protein chemist, wanted to pursue what are they? And it turns out they are cleavage products of UPF1. And doxorubicin is one way to generate these cleavage products. They are underrepresented here in intensity relative to full-length UPF1, this key NMD factor, because the antibody reacts with the terminal amino acids, and these cleavage products are lacking the amino terminus of full-length UPF1. And this work was done in human breast cancer cells. Max saw these doxorubicin-generated products, cyclohexamide-generated products, etoposide, another chemotherapeutic, in all cells that he looked at, cells from dogs, cells from mice, cells from cow, everything he looked at. And here's an example of mouse myoblasts, where they're given cyclohexamide or etoposide, which induces DNA damage. And again, you can see the cleavage product here. And this cleavage product occurs before the complete initiation of apoptosis. So it occurs before complete cleavage of the initiator and what do you call the other type of caspases? Initiator, and there's two types of caspases. Effector caspases. So this is part of the apoptotic process. It's not a consequence. It promotes, it leads to, it causes, feeds into apoptosis. So we know that UPF1 is cleaved by caspases because we can inhibit cleavage with caspase inhibitors. And to cut to the quick, Max mapped the primary cleavage site to an aspartic acid at position 37. All right, so there's a picture of UPF1. It's an ATP-dependent RNA helicase. Cleavage is occurring to knock off the first 37 amino acids of the protein. And what Max showed is that the level of UPF1 cleavage product, which he can transiently introduce into cells, will inhibit NMD more the more it is introduced into cells. And the level that we generate using doxorubicin is the level shown by the red arrow. And what we're measuring, Max is measuring, is the level of a PTC containing mRNA, the level of which goes up when NMD is inhibited. So this is a sub-stoichiometric cleavage of UPF1 that is certainly not inhibiting NMD as efficiently as could be inhibited if it were produced by cells in response to chemotherapeutic agents, if there were higher levels of it produced. So how do you separate all the stuff that happens when you damage DNA from the NMD aspects of the response? And so what Max did is he generated a variant of UPF1 that was fully functional, but it had, instead of the ability to be cleaved by caspase, a variation in its sequence so that Max could control its production by introducing a proteolytic site for the tobacco etch virus protease. So he could express full-length protein that was caspase resistant and then cleave UPF1 independently of any DNA damage and generate the same amounts of cleaved UPF1, the same structure, cleaved product of UPF1 as was generated with doxorubicin treatment. And then he sequenced the transcriptome that resulted. And this is a very short list. What he obtained were natural NMD targets that were upregulated and that promoted apoptosis. So this is growth arrest and DNA damage response gene encoding a protein that induces G2M arrest in the cell cycle. And there's Gas5, which is an NMD target, and its expression causes growth arrest. There's a whole bunch of NMD targets that promote apoptosis. And Max corroborated these data by using mRNA-specific and pre-mRNA-specific primers and RT-PCR analysis. So we can often attribute a reduction in mRNA abundance to an mRNA decay pathway if pre-mRNA levels are unchanged, but mRNA levels are. Right? Pre-mRNA is a precursor to mRNA. And Max also measured half-life. Also, coming from these studies, Max found that inhibiting UPF1 cleavage decreases and slows doxorubicin-mediated toxicity, which is what you would expect if the cleavage reaction promotes apoptosis. And he found that if the cleavage product were expressed or NMD was inhibited, that would speed up doxorubicin-mediated toxicity, as if NMD primes cells for apoptosis. If Max inhibits NMD, he doesn't cause apoptosis. But inhibiting NMD, by inhibiting NMD, the cells are pushed towards the apoptotic pathway. And Miles Wilkinson has shown in the case of differentiating neuronal embryonic stem cells to neurons, NMD is inhibited. And that, if you inhibit NMD, you don't promote neurogenesis, but together with all the other stuff that's happening during the differentiation of neuron process, the inhibition of NMD promotes neurogenesis. It's not a caspase-mediated cleavage of UPF1 in neuronal differentiation. It is the upregulation of a microRNA that downregulates the level of UPF1 mRNA. So cells have responded differently and inhibited NMD in different ways to promote changes in their metabolism depending upon the stimulus that they see. So I think some of the best work that were known for uncovered the mechanism of NMD, and I just want to mention that briefly. I've already told you that NMD occurs largely during what we've called a pioneer round of translation that utilizes mRNA that's bound by the cap binding protein heterodimer that typifies pre-mRNA. And this newly synthesized RNA, if it underwent splicing, will contain the exon junction complexes upstream, 20 to 24 nucleotides upstream of exon-exon junctions. This is deposited in a sequence-independent but position-dependent way. Here's the premature termination codon. Here's the ribosome translating that RNA. And what happens is when the ribosome meets the premature termination codon, provided the terminating ribosome doesn't physically remove the EJC, hence that 50 to 55 nucleotide rule, then the two normal translation termination factors will be joined by UPF1, the key NMD factor. It's an ATP-dependent RNA helicase, and its kinase, SMUG1. This is a complex that we didn't define, but it certainly exists. It was defined by Shige Ono's group, and it's called SIRF for the different components that make it up. And then UPF1 and its kinase move to the EJC, at which point its kinase phosphorylates UPF1, SMUG phosphorylates UPF1. There's a remarkable step of translational repression that I will tell you about that's critical for the mRNA decay step, and then the mRNA is degraded. RNAs in cells are never free of proteins, and what we've elucidated is the choreography of events on an mRNA with regard to how the proteins are moving around on it in a regulated way to mediate the regulated phosphorylation of UPF1, which is the trigger for NMD. Once the trigger is pulled and you get the translational repression step, the mRNA will be degraded. Olaf was a postdoc in the lab who understood that UPF1 phosphorylation triggers translational repression, and we did this work in collaboration with John Hershey, who is an eukaryotic initiation factor 3 expert. Olaf showed that phospho-UPF1 interacts with the two highest molecular weight subunits of this multi-subunit initiation factor, translation initiation factor, and that results in the inhibition of the 60S ribosomal subunit joining this 43S pre-initiation complex poised at the initiation codon, ready to go, ready to accept the 60S subunit to form a translationally active 80S ribosome. And phospho-UPF1 also directly or indirectly recruits degradative activities that we know function in NMD. And they include an endonuclease that I'm not going to go over called SMUG1, and enzymes that remove the cap and degrade the RNA in the 5' to 3' direction, or that remove the poly-A tail and degrade the RNA in the 3' to 5' direction. If we initiate translation from an internal ribosome entry site that is from a virus like cricket paralysis virus, internal ribosome entry site that is independent of EIF3, then even though that mRNA has a premature termination codon, it will not be targeted for NMD because there's no way to repress reinitiation events, ongoing translation. When we found that newly synthesized mRNA could be translated, we were really surprised. All the textbooks said that proteins are made from this mRNA with eukaryotic initiation factor 4E at the cap. And it's true, most of cellular proteins are made from this mRNA. However, it's easy to see why this mRNA is targeted for NMD, and by the time EIF4E replaces CPP80 and 20 at the cap, the mRNA is largely immune to NMD. Fabrice Lejeune was a postdoc in the lab who showed a precursor-product relationship between the two, and this RNA is immune to NMD because there's no EJCs, they're gone. And also CBP80 promotes NMD. And I just want to show you that was work by Jungwoo Kwang, who was a postdoc in the lab. UPF1 associates with CBP80 at the cap and promotes NMD at least 2 steps. All right, you've already seen this before, except now, look, UPF1 has a transient or weak interaction with CBP80, and that CBP80 escorts UPF1 and its kinase first to the termination complex and then to the EJC. In that way, CBP80 promotes NMD. So this newly synthesized mRNA with its associated proteins is set up for surveillance. And if there's a premature termination codon that doesn't remove downstream EJCs, then the mRNA will be targeted for NMD. Where in the cell does NMD occur? And I think this is the last thing that I'm going to tell you. How are we doing on time? Okay. You know, you can imagine I got a lot of pushback when I said introns and pre-mRNAs determine which termination codons in the spliced product influenced mRNA half-life. And I also got a lot of pushback by saying that NMD was largely nucleus associated. And I never said there was translation of the nucleus. And for many, many years, I would go to Stockholm. Bertil Denholt works on the Balbieni ring granule in Chironomus. It's an insect. It's a large RNP. It's about 45 kilobases that he could see using electron micrographs. And I always imagined that what we were looking at when I talked about nucleus-associated NMD, meaning that the mRNA co-purified with the nucleus in which it was synthesized, but could be translated by cytoplasmic ribosomes at a point where it was still co-purifying with the nucleus. Nothing heretical, really. I always pictured what Bertil saw, and that is for this large RNP, RNA-associated proteins, it's an mRNA, you could see ribosomes, these blobs, associated with the mRNA that was invariably exported from the nucleus to the cytoplasm 5' end first. And before the 3' end had yet to transit the nuclear envelope, so these two membranes are the nuclear envelope, and this is a pore through which RNAs go out of the nucleus. You see the 3' end is still associated with the nucleus while it's being translated. Here's another picture, and here's a spread out version, and here's my lovely drawing. And in fact, that seems to be the case, and we were finally able to show this, to visualize this by collaborating with Rob Singer, who's at Einstein. Hana was a postdoc in the lab. She had money from the Japan Society for the Promotion of Science, and she worked physically in Rob's lab with Tatiana, who was a graduate student at the time, and they were able to demonstrate where nucleus-associated NMD occurs in human cells. Remember how I told you that NMD is biphasic? Well, first of all, we just inhibited transcription using actinomycin D and drove globin gene expression with a premature termination codon or not. So here's the termination codon or not. This is normal. Using a FOS promoter that's transiently inducible when you add serum to serum-deprived cells. And the best fit curve for the decay is biphasic. So here's where NMD is occurring very early on. And when that RNA rearranges so it doesn't have CBP80 at the cap anymore, it has EIF4E, it's lost its EJCs, whatever has survived, NMD is never 100% efficient, is degraded with a half-life that is like its normal counterpart. This is what we saw by fractionating cells and by doing similar experiments. All right, so what we did with Rob is to use fluorescent in situ hybridization of globin mRNA that was PTC-free or PTC-containing, norm or ter, and we gutted the 3' and translated region with E. coli beta-galactosidase sequences, either A or B. And we drove the norm and ter, the normal and the PTC-containing mRNAs, from a bidirectional promoter. So we introduced them in the same plasmid, and the promoter was Pane-inducible, so we could synchronize the burst in transcription and mRNA generation. By looking at a cell, and the boundary between the nucleus and the cytoplasm was defined by DAPI staining, which stains DNA. So here's the boundary. And looking at the ratio of, what are we doing here, of PTC-containing mRNA to its normal counterpart, there's a lot of bounce, but basically there isn't a change in that ratio in the nucleus. There isn't a change in that ratio in the cytoplasm. But where you see the drop in the PTC-containing RNA is on the cytoplasmic side of the nuclear envelope. So picture what I showed you was happening with the Balbiani ring granule. And the green localizes the nuclear pore complexes that spot the nuclear envelope. We have an antibody specific for them. So NMD is occurring within, let's see what the next slide says. Oh yeah, so there was no detectable change in the ratio of the two in the nucleus or the cytoplasm. And our estimates are that it takes anywhere from 5 or 6 seconds to 56 seconds, so less than a minute, once a PTC-containing globin RNA makes it to the cytoplasm for it to be targeted for NMD. So it's while it's associated with the nucleus in which it was synthesized. And I'm not going to tell you that. All right, so what did I tell you today? So I told you that NMD is an important quality control pathway in human cells and all mammalian cells and all eukaryotes. It eliminates mistakes routinely made by cells when they express genes. It protects carriers of a single PTC-containing disease-associated allele where the other allele is normal from disease. NMD is also used by cells to autoregulate many RNA binding proteins in a process called alternative splicing coupled to NMD. NMD additionally targets 10% of our normal physiologic mRNAs, and it's regulated by cells as an adaptive process. And there are many examples. I talked about Max's work on apoptosis, and I mentioned Miles Wilkinson's work on neuronal cell differentiation. Viruses like to downregulate NMD. Otherwise, their replication is inhibited. There are many ways by which cells respond to their changing environments by regulating the efficiency of NMD as an adaptive process. And what did we uncover? That nuclear processes of mRNA formation can mark an mRNA in a way that affects how it's metabolized in the cytoplasm. We discovered the pioneer round of translation for the purpose of cellular quality control. We defined nucleus-associated mRNA translation, and I'm not going to talk about related pathway. That's a whole new regulatory ways that uncovered new roles for long non-coding RNAs. And with that, I want to thank you for your attention and acknowledge our funding sources. I've acknowledged people as I've gone along, and thank you.